Good morning. So today I will discuss about gene transfer in prokaryotes. Gene transfer is a transfer of gene from one DNA molecule to the another DNA molecule. It can be vertical or horizontal. In vertical gene transfer, transfer of gene from parents to offspring, and in horizontal there are transfer of gene between two independent organisms. Their main motive है gene transfer का कि first of all gene transfer होना one organism to another, then integrate करना उसके DNA में, then express करना. So transfer gene को हम transgene बोलते हैं. Then organism that develop after successful gene transfer is known as transgenic. There are three methods through which gene transfer takes place: conjugation, transformation, and transduction. But there are some artificial methods by which gene transfer takes place. Physical method है micro injection, biologistic transformation. Chemical methods DNA transfer by calcium phosphate method, liposome mediated transfer, and third one is electrical method, electroporation. First of all, we will discuss about conjugation. In 1946, Lederberg and Tatum ने शो किया कि बैक्टीरिया अंडरगो conjugation. Conjugation एक process है by which one bacteria transfers genetic material to another through direct contact. In conjugation, one bacteria act as a donor and another act as a recipient. Donor bacteria have fertility factor F plasmid, and recipient have there is no fertility factor. So F factor allows to uh, formation of pili. So they are connected to each other, formation of conjugation bridge through which gene transfer takes place. So through the conjugation tube, transfer of gene from one bacterial cell to another bacterial cell takes place. We have to you have to see in this figure bacteria cell F plasmid. Plasmid is present in donor cell, and F plasmid is absent in recipient cell. First of all, they form bridge between two cells, and then transfer takes place, and at last both the cell have F factor. Okay, then discuss about transformation. Transformation was discovered by Griffith in 1928. Transformation is a process of taking up extracellular DNA strand of one bacteria by competent bacteria cell. जो एक बैक्टीरिया डीएनए को अपटेक करेगा, वो भी फ्री डीएनए को, उसे हम ट्रांसफॉर्मेशन बोलेंगे. देन इट कैन बी नेचुरल एंड आर्टिफिशियल. मीन ट्रांसफॉर्मेशन और नेचुरल भी हो सकता है, आर्टिफिशियल भी हो सकता है. और द ट्रांसफॉर्मेशन occurs mainly in closely related species. Then you have to see in this figure. First of all, bacteria have DNA fragments. They are released into free space, then uptake by another bacteria cell, and then integrate and then express their gene. Last one is transduction. Transduction is the mode of genetic transfer from one bacteria to another through a virus. There is no direct contact between the cells. In this process, bacteriophage, which infect bacteria, use host cell to multiply, and while assembling this, sometime pack to bacteria DNA with them, and then these virus infect new bacteria and then transfer the bacterial genome to that bacteria, and then bacteria insert that DNA segment and express it. So transduction was discovered by Zinder and Landerweg in Salmonella. So you have to see in this figure. First of all, bacteriophage infect the bacteria cell, and then break down their DNA material, then packed into a phage, and then that infect the another bacterial cell. Then you have to see in this figure integrated into the bacterial DNA. So there are three methods of genetic transformation in gene transfer in prokaryotes: transduction, transformation, and conjugation. So this is, thank you.